So our last speaker of the day, a little bit late, but not too bad, I hope, Matt, is Matt Greving, who's another Neil Woodbury alum and has worked for several biotech companies, I think straight out of grad school, I think you did that. And I think you worked for HealthTel, and
Yep.
one you founded, Rubric. So
I founded a few companies,
yeah, so presumably we'll hear a little bit about this.
Yeah.
Thank you, Matt.
Thank you, Stuart. Thanks for everybody for sticking around, and appreciate the opportunity to celebrate 20 years at Biodesign. It really kind of set the stage for me to start many of these companies and actually do some quite significant things in the field of therapeutics. And without Biodesign, I don't think any of that would have happened. So actually not that long ago, close enough that my left foot, my frozen toes on my left foot are still only like 75% healed. In June, I climbed the north face of Mount Everest. And let me see, if you're interested, this is the Tibet side. This is the side nobody climbs. So it took me 3 years to get permits from China to do this. And actually this year, after I had all my travel booked and everything, they said, psych, nope, you're not going. And then they called back a week later and said, just kidding. So you can come. And I ended up spending an exorbitant amount of money to get last minute flights into Tibet to go climb the north face. And just for reference, the line that we climb is basically up this face here. And you can see there's a little bit of a notch there. And we just follow the ridge all the way to the top. And we have to come down. And then coming down is way harder. So I'm going to talk about why turning science into biotech ventures is actually quite similar to climbing that damn mountain. So like I said, I consider Biodesign an ideal training ground for what I did to build some of these companies with capable co-founders, of course. And this is me in 2006, actually just down the hallway. And I was weighing out amino acids to build these designed peptide arrays. And, you know, I was a computer scientist before I came in here, and when Neil Woodbury told me we're gonna basically do digital chemistry to look at the immune system, I couldn't turn it down. So that's why I turned down other graduate opportunities and came to Biodesign. And this idea of just measuring everything at scale is gonna be a consistent theme throughout my career. And so, you know, my dissertation was on how do you utilize this information from these peptide libraries to optimize peptides. And I was like super fascinated about, you know, even doing peptide drugs. Unfortunately, I didn't know about GLP-1s at the time because we could have been the ones to do semaglutide, but we missed that opportunity. Anyway, my dissertation was on how do you look at these design libraries and utilize that data to optimize peptides. And this works. I actually use it to this day to do antibody engineering, the concepts that we learned on this. And, you know, I had no idea that this is actually more relevant today with AI than it was back then, because this is exactly, I mean, we were doing additivity, but AI used similar concepts in combinatorial space. So, okay, I graduate and I'm at base camp. So the equivalent to getting to Everest is 4 days of flying, a 3-day Jeep ride, and a 1-day trek into this base camp at 17,000 feet. And you think, man, everything's awesome. I've got my PhD, but looking back, you're way far away from the summit of Everest and getting a successful biotech off the ground. So I went to Scripps and actually followed in Trent Northern's steps. And I'd seen what he had built with this high throughput or this silicon substrate for doing mass spec. And we had thought a lot about how could we utilize that. And we basically came down to, we could basically do high throughput label-free mass spec screening for things where like fluorescence just doesn't work. And so I filed for some SBIRs, and I ended up getting 3 SBIRs to form a company called NextVal. And the idea was that we could take the chips that Trent and others at Scripps built and integrate it with an acoustic deposition device that would deposit these nanoliter sample volumes, and you could look at, tens of thousands of samples and scan that with mass spec and basically do high throughput mass spec screening. And I had no idea, how anybody would use this, but I knew that if we could generate this kind of very high content data at scale, there had to be some applications. So I actually went to Cambridge, Massachusetts, where all the pharma companies are, and I tried to get meetings with them for months, and then finally got some meetings with them in February. Forget what year it was. It must have been 2012. And I realized when I got there why I got meetings with them in February, because Boston in February is very cold. And so nobody was there. In fact, I got like $80 a night hotel rooms at the W Hotel, which is like normally about $400 a night. So anyway, it was great because I got to spend about a month in Cambridge, Mass, working with the scientists at Novartis, Bristol Myers Squibb, Pfizer, other pharma companies, Vertex. And ultimately, Bristol Myers Squibb said, you know what? We have hundreds of thousands of these compounds, but we don't know their liver tox. We don't know which cytochrome P450 enzymes they inhibit, and we don't know what the metabolites of those drugs are once they're processed by the cytochrome P450s. We think this would be perfect for that. So I built this technology with Bristol Myers Squibb to do this high throughput liver toxicity and metabolism screening for small molecule drugs. And that ended up the following year winning the SLAS, just kind of the premier pharma screening organization, the Innovation of the Year award there. I think the key takeaway here is that, if you really have to listen to your customers because you're so deep in the science that you may not know the killer app. And getting out there and getting in the trenches, you can find those killer apps for your science. And there was another one actually working a little bit with Trent, really initiated, where credit's due, with the company Verinium in San Diego, which ultimately got acquired by BASF. And they said, you know what, this is really good for high throughput industrial enzyme screening. And so they made industrial enzymes, and we built this technology to do industrial enzyme screening as well. And actually, along again with Trent's help, we actually won the R&D magazine, one of the Oscars of innovation that year for this technology. So after 6 years, my business partner bought out the high throughput drug tox screening technology, and then BASF actually licensed the high throughput enzyme screening, and I didn't know what I was going to do, but I knew I was exhausted. And you can see me here at 25,000 feet camp with my climbing partner Carla. I'm wiped, and I'm still a mile from the summit. But she's laughing. Why is that? Well, this is my first time on Everest. This is her third. And my first startup was way tougher than I expected. And just like climbing Everest the first time, you can't anticipate how hard it's going to be till you actually do it. She's laughing at me because she knows where she was her first time. And just a side note, Carla is the first woman to climb Everest and K2, the second highest peak in the world, without oxygen. She used oxygen this time, but the prior time she didn't use oxygen. So experience builds up what we call scar tissue, startup scar tissue, and things bother you a lot less your second time. But I was sitting there, basically had exited NextVal, not knowing what to do, and then I got a call actually from HealthTel, and I was, again, never lost that love for looking at peptide interactions at scale. So we started studying peptide antibody interactions at 3 million peptide scale on these photolithographic peptide arrays. And something interesting came out of looking at that data at scale. There's a lot of work behind this, but basically what it came down to is that we discovered, and this is before deep learning was even popular, we discovered that if we converted these protein structures, this is PD1, this is real data that we actually did, if we converted these structures to graph networks, we could take the large-scale peptide binding data to the antibodies that bind PD1, and we could start to align them to this graph. And once you align it to the graph, you can convert that graph back to the three-dimensional crystal structure in this case, and you can basically map where did those antibodies bind onto the target, in this case PD-1, that's nivolumab's binding epitope. And this got kind of interesting because I started to think, well, can we do this backwards? So if we know we want to bind a target or an epitope on a target, could we design a sequence, a peptide sequence or a small mini protein sequence that adopts a structure that would present the structural epitope of the target and then use that effectively to steer an immune response towards an epitope for antibody discovery? And that insight actually convinced some investors for us to raise money to build a therapeutics company. So that made me realize, like, these unique insights, when you've seen something that no one else has seen, nobody believed that. And investors actually forced us to do, well, I shouldn't say forced, but asked us to do a blinded study with Merck where they gave us 13 antibodies they had in discovery. They knew the epitopes they bound to. We had no idea. And we got 11 out of the 13 right. And so that's what effectively triggered the funding, that yes, we can do this. So when you see these unique insights, you should think about, like, is there an opportunity in that? So this is 4 o'clock in the morning on I don't know what day I summited, but this is 4 o'clock in the morning, and it was very surreal. I realized we had that whole mountain to ourselves. There's only 4 people in the world that saw the sunrise at that elevation, because at 28,500 feet, I'm higher than K2. So there's no other person on the planet that saw the sunrise at that elevation other than myself and my 3 climbing partners. So these unique insights can be real opportunities if you recognize them. So we got funding from venture capitalists to form Rubric Therapeutics, where we took those insights and focused on developing antibody therapeutics. So the idea here was that we'd have a target protein, and these would be targets where traditional antibody discovery was very difficult. And this is actually CCR8 GPCR. It's actually a molecule that's ready for clinical trials right now that came out of this. But you could design, like I said, these peptides that could adopt these structures, and then you could mix that with an antibody library or immunize a mouse, let's say, with these structured epitopes. And what would come out are epitope-specific antibodies. And this worked, I mean, unbelievably well. So, we got a number of therapeutic campaigns out of this. I think a total of probably on the order of 6, with 4 molecules in development right now. And one thing we realized, though, is that when you get these antibodies out, a lot of times you have to optimize on a number of dimensions. These aren't drugs. They require a fair bit of optimization. So we actually, again, just kind of went out there and tried something that no one else had tried. This is in 2017, before deep learning and proteins was like huge. People were still skeptical. So we took millions of human antibody sequences. We trained an AI model, and I'm actually showing the real data. This is what's called an attention map, where if you just map, how much one position knows about another position, at least the model has learned, you start to see some patterns in these large-scale data. And what was very obvious to us, because we had stared at so many antibodies for a while, is that you can see that there's high attention in these intervening regions and low attention in the CDRs. It makes sense, because our CDRs are the hypervariable regions of the antibodies, so the information content is a lot lower in those regions. It's not obvious how you would use this at the beginning, but what we realized is that this AI model, if we combine it with some screening data, meaning we give the model as input some sequences and give it the corresponding activity of those sequences, what would come out are optimized antibodies that had significantly better activity on whatever dimension we were optimizing on. And we kept the secret, and we probably shouldn't have, because about a year later, someone published it and got huge accolades, and we're like, wait a minute, we did that a year ago, but our fault, we kept it secret. But long story short, that was our platform. We would epitope steer and then optimize very rapidly those hits into drug-ready molecules, antibodies, using that AI model. So I'll show one example because this leads into my next company that I'm working on now. We went after, those of you who saw Carolyn's talk last night might recognize this, PD-1, but we're doing something different. We wanted to treat autoimmunity. PD-1 is a checkpoint inhibitor, so when you activate it, it suppresses the immune response. Autoimmunity, the immune response is overactive. So let's agonize or activate PD-1 and suppress the immune response in autoimmunity. This is a very new idea then. Now it's one of the most popular autoimmune target mechanisms out there. We designed a peptide that actually was steering the antibodies towards an agonist epitope on PD-1. We got some PD-1 agonist antibodies, which are very difficult to get. And one of the problems is that when we get these antibodies, yeah, we saw they bound, but again, we don't care about binding so much. We want to see, do they activate the pathway? And ultimately, this took us a year of just brute force, low throughput screening in vitro assays to find some PD-1 agonists. And ultimately, we just sold the molecules to Otsuka Pharmaceuticals last April. Ultimately, we did find a number of very strong candidates. These are the clinical benchmarks. This is Bristol Myers Squibb's molecule. This is a company called AnaptysBio, which is in phase 1 or maybe even phase 2 now. This is the natural agonist that we all use when we don't have autoimmunity, PD-L1. You can see two of our leads actually basically match the immune suppression activity of the natural agonist PD-L1. So these molecules are actually being developed by Otsuka right now for autoimmunity. Like I said, going after these difficult mechanisms, these difficult diseases like autoimmunity or solid tumors is another example of a complex disease. That screening was so low throughput. I mean, we about killed ourselves just to find those molecules. And that was a major obstacle for us. So we said, okay, we need to build something to fix this problem of complex disease, complex mechanism screening at scale. So this is 28,250 feet. This is the crux of the north face of Mount Everest. And those of you who know climbing grades, it goes at 510. These ladders were carried up in 2007. This is us setting up the ladders so we can get through the really sketchy parts. And yeah, this is the major roadblock on the north face of Everest to the summit, and overcoming that reveals the access to the summit. So we had to build something, and this is my last company that I'll talk about today because it's my current company. I bootstrapped this one out of my own pocket to get some proof of concept data, and now we're actively engaged with venture capitalists to fund this. But the idea is that we can do complex mechanism in disease microenvironment screening at scale, so 100,000 scale, and we're measuring everything. So in each of these nanowells on a chip is a complex disease microenvironment intending to represent, let's say, autoimmune conditions. So there's multiple cell types in here. And then from these, we can take imaging readouts, so anything that you can image, we can get a functional readout, and then we get sequencing, full transcriptome sequencing actually from each of these wells. So we've shown actually now in a couple proof of concept studies, actually even have an asset that we generated, that this works very well, where we can take antibody secreting cells, we use CHO cells, then mix them with these disease representative cells. This could be representing a solid tumor microenvironment or an autoimmune microenvironment, load that co-culture of cells into these nanowells, and then screen for functional hits. Stoic Therapeutics is focused on two modalities of drugs, antibody drugs. The first is antibody drug conjugates, where we're delivering payloads to target cells and hopefully not to the off-target cells. And then we also have in development some immune cell engagers where you're engaging an immune cell on one side and a target cell on another side to mediate some activity. So complex mechanisms, complex disease environments. And like I said, we're right now in massive fundraising mode, so excuse the fatigue in my voice. I've been up late at night actually pitching to investors in in different parts of the world. So startup lessons learned. This is going to be hard whether or not your problem's small or big, so you might as well go big. So just there's a baseline effort to start a biotech startup, and that baseline is hard. So make sure you're solving a big problem that people will care about. This is me on the summit, 29,029 feet on Everest. And the other key takeaway that I absolutely realized and will not back down from is that the team is the key to the success. You could have the best idea in the world, best technology in the world. If you don't have the right team, it's not going to succeed. So this is the team that I went up on Everest with, and this is us safely back at base camp with the summit of Everest in the background there.
Well, I'm glad you made it back, Matt, or we'd have had a problem. Questions for Matt? Well, I think we're...
No, there's one over here.
Oh, we have got one. Okay.
Go for it, Trent.
Of course, yeah.
We have to record.
You showed that picture, it was like a couple images ago, and there looked like there were a bunch of harnesses or ropes up on top of them. What's
Oh, yeah,
that
no,
about?
you have to put up
Right
ropes.
there. Oh, no, go back. There was just...
Oh, the on the summit.
Yeah, what's with that?
Oh, these are prayer flags. Remember, prayer flags. So this
Wow.
summit remembers at the triple point of Tibet, Nepal, and India. Very Buddhist region. Prayer flags. Prayer flags.
Oh, prayer flags.
Yeah.
Okay.
So all the Sherpas, even the Sherpa I was with, they carry up prayer flags.
Wow, thank you.
Yeah.
Thanks for asking, Trent. I wanted to ask.
Yeah.
It's,
And I will tell you that it's about a 7,000-foot fall on my left or on my right side. So if I fall off this side, that's a bad day.
okay, so I think this was a great note to end on. We started the morning with Space Camp,
Yeah, nice.
and we ended the day with Base Camp.
There you
So
go.
thank you.
Perfect.
Perfect, Coda. Thank you all for coming.